Welcome to our virtual lab tour. I'm so excited to be your professor today for this explainer. We're going to dive right into the super fascinating behind-the-scenes world of our animal assistants. These are the amazing creatures that actually help us create the life-saving medicines we rely on every single day. Now, I want you to think about this for just a second. When you or maybe a family member gets sick and you pull a pill bottle out of your medicine cabinet, how did that medicine actually get there? I mean, how do we know it's going to cure a headache and not just give you a terrible stomach ache instead? Well, it's actually a massive, incredible journey from a laboratory discovery all the way to that little plastic bottle. And, as you'll see, it relies heavily on some very special helpers. Okay, let's dive right into this. Here is our syllabus for today. First, welcome to the lab. 2, meet our animal assistants. 3, how do models work? 4, testing new medicines safely. And finally, number 5, the future without animals. All right, starting off with section 1, welcome to the lab. We're going to take a quick look at the history behind all of this. You know, scientists haven't just started working with animals recently. This is a practice that goes back thousands of years. Just look at this timeline. Way back in ancient Greece, Aristotle actually studied animal anatomy just to understand how living things are put together. It's like a giant puzzle. Fast forward to the Renaissance, and William Harvey used animals to prove that the heart actually acts like a mechanical pump for our blood. Then, in the 19th century, brilliant minds like Louis Pasteur developed the very first vaccines using animal models, which basically proved the germ theory of disease. And then in the 20th century, boom, a massive explosion of medical miracles. We're talking about things like insulin for diabetes and penicillin to fight off infections, and all of those were discovered and proven safe in animals first. Okay, moving on to section 2, which happens to be my absolute favorite part, meet our animal assistants. Now, what's really interesting about this slide is our class roster. You can see a huge variety here, right? Everything from macaques to hamsters. But I want to draw your attention down to the bottom left, the mouse. Mice are literally the Swiss Army knives of the laboratory. And why is that? Well, because they're small, they grow incredibly fast, and believe it or not, their genetics are surprisingly similar to ours. We've actually mapped their entire genome. That allows us to study everything from cancer to obesity right there in that tiny little assistant. But what if a mouse is just a bit too small for what we need? Let's check out the data on the chalkboard for our next assistant, the rat. Rats are a bit larger, living about 1 to 3 years, and get this, they have a wildly fast heart rate of over 300 beats per minute. Because they're bigger and they actually have a really calm temperament, they are absolutely perfect for tests where we might need to draw just a tiny bit of blood, or when we need an animal that's relaxed enough to figure out behavioral puzzles. We also use some highly specialized assistants. Take a look at this comparison between the zebrafish and the beagle dog. Zebrafish are practically magical. I mean, their embryos are completely transparent. Scientists can literally look right through their skin like a clear glass window and watch their tiny organs develop in real time. It's amazing. Beagle dogs, on the other hand, share deep anatomical similarities to human organ systems, especially the heart. Because of this, they are uniquely suited for testing heart medicines. Though, of course, their use is highly restricted and closely monitored for ethical reasons. All right, let's jump to section 3. How do models work? So, the crucial point here is really understanding this concept of a model. A model is basically just a way to recreate a human disease inside an animal. And one of the most brilliant ways we do this today is through what's called a transgenic model. This just means scientists engineer an animal with foreign DNA to perfectly copy a human genetic disease. To do this, they use a mind-blowing technology called CRISPR. You can kind of think of CRISPR as microscopic genetic scissors. These tiny scissors let scientists carefully cut and paste DNA so a mouse can be born with the exact same genetic mutation that causes a specific disease in humans. It is incredibly precise. But transgenic is just one type. There are 4 major ways we model diseases. Sometimes we use induced models, where scientists actually trigger the disease safely. Sometimes we use spontaneous models, which is when we find an animal that naturally developed a genetic mutation on its own. We also have humanized models, which sounds crazy, but it's where human cells or even human immune systems are put into a mouse. And finally, infection models, where we expose the animal to a bacteria or a virus so we can test new vaccines. Let's look a little closer at induced models because they're super common. Imagine we're trying to cure type 1 diabetes. We can't really just wait around hoping a rat naturally gets diabetes, right? Instead, scientists use a specific chemical with a very long name, streptozotocin. This chemical safely targets the pancreas in the rat and triggers a diabetic state. So now, the rat has diabetes, and scientists have this perfect, highly controlled environment to test a brand new insulin medicine. A medicine that might end up saving someone in your own family. Okay, moving on to Section 4, testing new medicines safely. And this brilliantly illustrates one of our absolute biggest challenges in the lab. Think about it. How do you ask a mouse if a new medicine is making it feel anxious or depressed? You can't, right? So scientists use central nervous system behavioral tests. Just look at these clever setups. If we want to measure anxiety, we use an elevated plus maze or a light dark box and simply see where the mouse prefers to hide. If we want to check social behavior, we observe how they interact with a completely stranger mouse in a clear box. It's fascinating. We can actually test invisible feelings just by watching their physical choices. Now, another massive part of safety testing is toxicology. Almost 500 years ago, a scientist named Paracelsus figured out a golden rule. The right dose is what differentiates a poison from a remedy. Think about it. Even drinking way too much water can make you sick. Toxicology uses our animal assistants to find the exact, safe starting dose of a medicine before a human ever even touches it. We have to figure out exactly how much cures the disease and how much might cause harm. And students, I really need you to pay close attention here, because history has taught us a very tragic lesson about exactly why this strict safety testing is absolutely mandatory. In the 20th century, a drug called thalidomide was given to pregnant women for morning sickness, but it completely bypassed thorough multi-species safety testing. Tragically, it caused thousands of human infants to be born with severe limb malformations. It's a heartbreaking historical event, but it's the exact reason why today, every single medicine must be strictly tested on multiple different animal species to ensure it won't unexpectedly harm human development. We literally cannot skip these steps. All right, that brings us to our final stop, Section 5, the future without animals. So in every modern lab, there are extremely strict rules enforced by ethics committees. They're basically the classroom rules of science, and they're called the 3 R's principle, originally created back in 1959. Rule number 1, replacement. Basically, if you can use a computer or a cell culture instead of an animal, you absolutely must do it. Rule number 2, reduction. Scientists have to use the absolute minimum number of animals necessary to get good math and good science. And rule number 3, refinement. This means scientists must constantly improve their procedures to reduce any pain, stress, or discomfort the animals might feel. Animal welfare is a huge priority. Let's move on and see how this builds into some truly sci-fi technology. Because of that replacement rule, scientists have invented some amazing advanced alternatives. We now have organ-on-a-chip technology. These are tiny little microchips lined with living human cells that act exactly like a mini lung or a mini liver. No way, right? We can actually 3D print living human tissue layer by tiny layer. We can grow cells in petri dishes, and we can use super advanced computers to simulate biology. And speaking of computers, artificial intelligence is completely changing the game here. We're actually teaching smart AI computers to analyze massive, massive amounts of historical data. These machine learning models are getting so incredibly smart that they can often predict if a drug is going to be toxic before an animal is ever even used. That is a massive breakthrough for that reduction principle we just learned about. And that brings our virtual lab tour to a close. But I want to leave you with a very big question to ponder. With a rapid rise of computer chips that mimic our organs, 3D printed tissues, and highly advanced artificial intelligence, will we one day be able to replace our animal assistants forever? It's an inspiring technological frontier, and who knows, maybe one of you sitting in class today will be the brilliant scientist who finally makes it happen. Thanks so much for joining me on this explainer. Class dismissed.